original voice. Oh my god. It took a long time for me to reconnect in Sotero. I am a singer, original voice in the house. Let's go back live streaming. Oh my god. Wait for a while. I have to reconnect my laptop to the net also. Hi, Johnny the Cricket Oval. Okay. And I'm very, very happy now. Okay. So my blood flow circulation is working very well. Okay. And then it's also very effective. Oh my gosh. Okay, thank you so much for that. Chain pairs, right? Oh my god. Really? Okay, wait for a while. Gosh. Wait for a while. I have to readjust the screen. Oh my gosh. This one. Okay. Molecular structural form. And I will be able also to illustrate the RNA, the ribonucleic acid, and nucleic acid sequencing or the genetic information. Okay, composed of nucleotides. These nucleotides, okay, nucleic acid sequencing, what we call as a genetic information of life, all living organisms. Okay, and this is also serve as a primary, messenger carrying molecules in cells for the protein synthesis, store, transmit genetic information of all living organism, of all life forms on Earth, something like that. Okay, so transmitting and storing genetic information through this protein synthesis. Okay, so protein also is very important in dealing with nucleic acids. So they are interrelated with each other. Okay, there's a relation between the two. This later I will be able to discuss, but that's not our topic today or this moment in time. Okay, after this we'll go to phospholipids, right? Of amino acids in protein is determined by the nucleotide sequencing of genetic coding, right, of DNA protein or protein DNA, right? So it's gonna be like that always. Okay, and then each nucleotide composed of nitrogenous base and H2 or H2 and reverse, okay? And then pentose, oh my gosh, happening, eh? Guys, pentose, 5 carbon atoms, and the phosphate will start to nucleobase pairs. Actually, nucleobase are discovered by Albrecht Kossel in 1880. Okay, he identified the nucleobases that has the Guanine, Uracil, and Thymen, like that. And after that, 1953, Mr. Watson and Mr. Crick, presented or demonstrated research through the aid of laboratory tests and experimentation, okay, by cytosine, okay. Actually, I showed that a while ago, right? But because I need to do my exercise and then come back now. And it took me a long time to reconnect again because there's a problem with my, connection in my laptop. I don't know why. Oh my gosh. So all my pentel pens are consumed just because of this. I have green pentel pen. I have blue. I don't have black. Always green. I don't have black. Always green. I love green because green represents environment. Clean and green. Oh my gosh. Like that. Okay. Let's start. Oh. So, hexagon. 1, 2, 3, 4, 5, 6. Like this. Okay. Like this and like this. Okay. Like that. Okay. And then NH. And this is NH2. Okay. This one, cytosine. This is the first nucleobase, or it could be arranged in no particular order. It depends on the the pairing of this nucleobase during the protein synthesis. Okay, to produce a genetic information or genetic instruction, something like that, for hereditary traits and inheritance, something like that. Okay, this is cytosine. Actually, we can represent this as letter C. Okay, this one, letter C for cytosine. Okay, I think biological students or best bio students can understand this. Okay, cytosine, and then we have the we have the uracil. Charot. Okay, this is the uracil. Okay, like that, and then you will take note also. It's gonna be like this. This is the uracil. Okay, beautiful, right? Look at orientation, huh? It will never be interchanged. Okay, uracil is applicable only to RNA. Review, what is the function of RNA? Okay, again, the specific sequencing for the parts, the structures, and the chemical reactions of all living organisms, or all. It has to do something with cells, because this new protein synthesis will be on the inside and out in the cells. Okay, like that, and then it can it can produced and encoded, okay, created, and then transmitted, and then be transmitted, but they have to express to really inside our body. Okay, so all of them are genetic information, okay, or genetic instruction. Okay, like that. So we have thymine. This is not thiamine, huh? Thiamine is vitamin B, right? Vitamin B and vitamin B3 is niacin, if you remember that one. This is for DNA alone. Okay, Deoxyribonucleic acid. You know the terms, the acronym, it's very difficult to justify, right? Because the acronym, it's very difficult to justify, right? Because deoxyribonucleic acid. So where's the N there? So N represents the nucleic, but the deoxyribonucleic is one word only. Okay, in English, if you put acronym, it should be each word, right? For example, what is the example? Science and technology. So ST, like that. What else? So many things, right? It's an acronym. Okay, truth. Okay, ASTM, American Society of Testing Materials. Okay, ISO, like this, International, okay, Organization for Standardization, something like that. So this one, DNA is very amazing because the N is continuously in the deoxyribonucleic. Deoxyribonucleic is one word only. Okay, and then acid, A acronym. I don't know because doctors are saying also DNA, but actually it's only DA because deoxyribonucleic is one word. You understand? Deoxyribonucleic is one word. Okay, and AA, and here also in RNA. So this is ribonucleic acid. Supposedly there's no N. That's also RA. Okay, because ribonucleic is one word. It doesn't follow the pattern of acronym. Okay, in English. It's English, right? Abbreviation, acronym, like that. Cutting off the words to make it understandable and easy to understand. But in here, it doesn't follow ribonucleic. Spelling, at least you know already what is the definition, what is the meaning of DNA, because we always heard that in the social media. We try to investigate the terms for that. Okay, for RNA, it's also ribonucleic acid. Okay, the ribonucleic acid, okay, actually that's RA because it's only one word, right? Ribonucleic. But I think because of the word nucleic acid, right? Nucleic acid, so in A. Okay, but it is really the rule for English, right? And ribo is the ribose sugar. This is very important raw material for protein synthesis in the RNA, the ribose sugar. If you can remember, I showed the formula for that. There are 5 carbon atoms for the ribose, right? You remember that one? That is the ribose. Okay, pentoses, under pentoses, under aldoses, which is a monomer of sugar, which is simple sugar. Deoxyribonucleic, that add only deoxy. Okay, okay, like that. Adding only deoxy. Okay, so, for the thymine, it's only applicable for DNA. It will never use the uracil. Okay, this is the only two nucleobase, okay, nucleobase that are specific for them during the pairing or the interaction during proteins. And after that, the thymine is like this, gene, right? Okay, this is the, this is the issue and the problem for biochemical engineering and chemical engineering, because mostly the subjects are through imagination. Imagination we cannot see in our naked eye. We're just imagining the protons, the electrons, the atoms, and we can never see that in our naked eye. The moles, the molecules, the cells, the proteins, any substances, the enzymes, the catalysts, the isotopes, the isomers, like that, the polymer, the monomer, those are biological substances, okay, or chemical substances that we can never see directly also in our naked eye. They are just functioning and regulating inside our physiological chemistry inside our body. And we can only identify their behavior, their characteristics, and their interaction through laboratory testing with the aid of new technology attached in the computer, something like attached with the microscopic devices, so they can see what's going to be the behavior of the bacteria, something like that. So that's why it's very important to vlog or to live stream. At the same time, you are inside the laboratory to really explain what's happening, the behavior and the characteristics of those various raw materials interacting with each other. Could it be the biological substances, the protein, the lipids, the fashion with the theories, principles, the basic concepts. We cannot back up with how they behave really in the actual scene inside our body. Okay, in the anatomy or structure of our body, and also in the physiological behavior inside our body, something like that. Okay, but this is only just to show, to interpret, so that we can understand a little bit that this is gonna be the behavior, the function, and the characteristics of the information and genetic instruction, something like that. That is the function of the gene, specific sequencing, meaning to say organized and properly arranged, okay, something, and pairing with each other, okay, something like that. Okay, so going back, grabe, in H, okay, I love really in H like this. So the difference only is attached, what do you call this one, a methyl group. You remember that one in a basic hydrocarbon, we have methane, ethane, methane, like that. We have acetyl bond. Okay, it will become triple bond if it's ethylene, ATHYLN, but that is acetylene, okay, together with oxygen gas cylinder, something like that, flammable gas cylinders, okay, like that. So if we remember that one, okay, this H3C, it looks like the reverse of, shall we say, alkyl group. Oh my goodness. Alkyl talaga. Hi! What's happening? Well, Uracil is your 2E. Actually, H3C is the same as CHE. Okay. like that. I will discuss a little bit with CH3, this one. CH3. So the alkyl, alkyl group. This is alkyl group in organic chemistry. We represented that as R. Okay. So if you remember the formula of protein, there is R there that will try to attach to the carbon and hydrogen together with the okay, alkyl group. Since alkyl, so this is there are alkane, alkene, EOO, like that. Vowel, right? The consonant are the S, T, B, U, V. In English again, okay? Consonant and vowel, okay? The alkane, look at my pronunciation. It matters a lot, okay? So, alkane. I just discussed a little bit in relation to that thymine, huh? Okay? With actually alkyne or alkane. What is the exact or appropriate pronunciation of this one? I forgot. Okay, this one. Alkane, alkene, and alkane. Oh my gosh. So this is the only consonant of the three alkyl group. So meaning to say the two are vowels. The letter E as an eagle for the double bond and A for the single bond. And for the triple bond, that is Y, which is the consonant Y. So we can represent that. We have also formula for that. Okay, the propane like that we use in our state of alkyl group banded together between carbon and hydrogen. So that is either covalent. Okay, a covalent bond is equal sharing of electrons, so non-polar covalent band. Because this carbon usually have, not usually, but by concept, it has 4 electrons in the outermost shell. So this will be divided completely to be bonded with hydrogen in the alkyl group, something like that. Okay, so it's gonna be like this. Example of an alkane is methane. Okay, methane. Example of an alkene, charot, is C2H2, acetylene. Okay. Example of alkane is the ethylene. It's a type of plastic, right? And we have also HDPE pipe, high density polyethylene. So the ethylene word there is coming from the alkane group that has a triple bond. Okay, that's the formula is C2H4. Okay, so density polyethylene. Okay, HDPE pipeline, something like that. Okay, that's come from this C2H4 chemical structure. Okay, number 3. And now we go back to the general formula of alkane, something like that. So CN. Let's okay. Let's consider the general formula, but we will just prove this. CH4. So if you use the formula of alkene, which is CnH2n plus 2, CnH2n plus 2, and N represent the number of atoms. So H4. Really? So this is the formula of for alkane. So we have methane gas. Okay? It ends with OL, that's alcohol, automatically. 1 carbon atom. But this is only an alkane. Okay? CH4 is methane. And 1 only carbon. So this N, it represents the 1. Okay? And 1, 2 times. This is correct of carbon atom. Like that. Like this. These are hydrocarbons, huh? Don't touch with anything. Exclusive for carbon and hydrogen. That's why we call this a hydrocarbon compound. Group is the double band. Okay? Double band, 2 underline, single band, 1 underline only, like this. Okay? So in the double band example, the acetylene, that's why we have acetylene gas. Okay, like that. The formula is CnH2n minus 2. So the carbon there, H2, 2, 4 minus 2, C2, 4 minus 2. This is CTH2, correct. Oh my gosh. My memory works so fast. Okay. So CnH2n minus 2 is the formula of alkene group or double band group in an alkyl group. Okay. These are all alkyl group subdivided into 3 groups. Okay. And then we go now to the triple band, which is the alkane. Okay. So if we put example as an alkane, ethylene, okay, as a hard plastic that cannot be biodegraded by environmental factors such as temperature, pressure, humidity, moisture, energy corrosion. Okay, there's a lot type of corrosion, right? So we have the cavity corrosion, the pitting corrosion, and the rusting corrosion through the reaction of ferric chloride, right, from water. Okay, no need to discuss that further because it's in organic chemistry. Okay, like that. But we go back to the ethylene. Okay, this ethylene is correct. C2H2. So C2H2 is example of alkene. Okay, and then C2, C2H4. Okay, ethylene. This one, sorry. C2H4 is ethylene. This one. Okay. So like this. So meaning to say, a multiple bands of polyethylene compose this as a monomer. Okay. Now, understood that of this ethylene C2H4, which is involves triple bond. Okay, like this. Okay, so this is very useful in our plastics. So we are just discussing the composition, okay, of those materials, construction materials. Okay, something like that. This is gonna be okay. The CH is the alkyl group. You have to attach that here in the thymine. Okay, H3C. Okay, something. There are 3 hydrogen and 1 carbon atom. Okay, but what is the name? The alkyl really? CH is the starting point of the alkyl. Sorry, that is the R. Okay, like that. Okay, so this thymine, and now we go now the adenine. But please don't mix the amino acids with the enzymes and the hormones, and then this, nucleotides, because they are basically the same. It ends with NE, NE, and sometimes ASE, something like that. Okay, you should identify. Okay, like that. For example, BS Chemical Engineering Board Exam Day 2. That is an essential examination, as always multiple choice. Let's say, for example, only like that. Okay, say that there is histidine, the valine, those are essential amino acids. You this one, this cytosine, uracil, thymen, like that, adenine, like that. That's why we have glycine, multivitamins, that is, amino acid. Okay? Like that. Mostly, those amino acids are found in the multivitamins also, or vitamins, okay, phenolics, because this is the raw material of antibacterial material to prevent and control cancers, okay, like that, cardiovascular diseases. They will insert light. Oh my gosh, ang lalim naman, metabolite talaga siya. Okay, an example. Okay, if you eat tomato too much every day, so there's a substance there that can reduce those cancers, cardiovascular diseases, viruses, and bacteria. That is very also important to eat tomato every day. Tomato, okay, the red color, the main source to fight, to control or prevent cancer, is a component of vaccine. Okay, AstraZeneca vaccine, Pfizer vaccine, or wrong. Okay, if you see the package of those vaccine, the trade secret products after by the government, right, in different countries before it will be sold to the market. So there must be a patent first. So there are all the something like that. Oh my gosh. Hello guys. Are you still there? What I am trying to say, that not being reflected in the nutritional facts due to the reason for trade secret, because maybe it will be copied by the other company. Before I work also in one of the soft drinks industry in the Philippines, the Coca-Cola Bottlers Philippines Incorporated. So we have an ingredients there known as concentrates. So this concentrates coming from Atlanta, USA. Okay, we have our chemists at the top. Okay, the soft drinks quality, blah, blah, blah. So, and so a lot of tests will be done. The chemist will be mixing there, but the proportion, they will wait for our computation before they will mix there upside. We will be the one to compute the proportion. It's gonna be like the sugar is like this, the carbon dioxide is like this, the sugar is like this, the concentrates is like this. So we compute, and then the chemist will receive that upside, and they will mix in a tank, something like this. That's their like that, and we will, after that we will sign, the analysis reports, and then we will tag QZ after releasing the products if it's passing the standards or the conformance, quality conformance specifications before releasing to the market. So it should be also having the patent, PATNT, that telling that outside, something like that, okay? So the concentrate is not mentioned there. If you buy a soft drinks, Coca-Cola, okay, Coca-Cola, it's never mentioned there, the breakdown. It's just put their concentrates. The concentrates are made of trade secret ingredients that nobody knows, so that it will not be copied by Pepsi, like that, because Pepsi have their own also concentrates of their own. Okay, though the procedure in making soft drinks is just totally the same generally, but there are a specific, what do you call this one? Taste or quality that coming from their own that it will not be copied to other companies. That is trade secret for the down of those ingredients. Okay, we call that as a concentrate. Okay, it will be delivered in our plant in a package like that, and then we don't know what's the composition for that also, because it will just arrive there as a concentrate. But we are sure there is caffeine, there is a CO2, there is water, there is sugar, and there is also phosphoric acid in that. That's why I always keep on telling you, okay, soft drinks have diluted phosphoric acid. Okay, if there is a phosphoric acid, it will add also to the phosphates that we receive in the adenosine triphosphate in the photosynthesis. Okay, that's why in our kidney, okay, there's also phosphates and phosphorus that will stuck in the gallbladder as a stone. Potassium, magnesium, calcium, a lot of salts will develop as a salt, and you will experience a kidney failure, and then you will be undergoing a dialysis procedure. Okay, something like that. That's why excessive intake of soft drinks is not good to the body. Okay, like that. Going back, my gosh, what is overconsumption is our enemy, and then less consumption is our friend. Just think of that. Okay, connotation, or saying, or a quote, QUOTE. Okay, like that. That is inspirational, motivational activity. Then it's okay. That is too much because it produces appropriate, okay, for the good health. What I mean is for the intake of the food, intake of the drinks, like blah 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 ones. Okay. My gosh. Going back to we touch back to adenine. Okay. My gosh. Hello guys, are you still there? You know if if you try to be strict, not strict, if you try to be detailed in your discussion, I think the students learn a lot. Okay? If and only if you have more time in your subject. For example, if I'm the instructor for like this, I'm instructor for General Chemistry 101, something like that, then I have to, explain it in detailed manner, then I think 1 hour or 2 hours are not enough. That's why maybe I will go direct to the most important part because time limited, something like this. Okay. Okay. Oh my gosh. Something is messaging me about the, iPhone. Oh my gosh, iPhone talaga siya. Fairness. Bening, aromatic hydrocarbon like this. 6 sides. 1, 2, 3. 6 sides. 1, 2, 3, 4. The same as benzene ring, right? Like this. But this is illustrating only the polygon, okay? And then there's also a double bond opposite to the other side, like this. Okay? Like that. Now you apply the technique. Okay? As I told you before. Okay, like that. So the technique is, if there's no double bond, you will put an H. Okay? Like that. Okay? Like that. So you have the NH2 at the top corner, like this. NH2, NH2 amino group, because this is a, this is nuclear base, right? Like this. And the double bond here, add nitrogen. Okay, because in every double bond you can put nitrogen. So see, I put nitrogen on the other side, right? Because every double bond you will put nitrogen in the corner, something like that. Okay, nitrogen here, nitrogen here. So the clue of adenine, the corner and the other corner is no attachment. There's no covalent band, something like this. In the corner, I will mark a circle like this. Okay, there's no covalent band in this corner, so that's your identification. Okay, and then we go to guanine. Charot. So actual information or genetic information for this, genes, functions, something like that. Okay, in our body, pairing, something like that. Okay, guanine, the same thing with adenine. Take note, only guanine and adenine have 2 polygons attached with each other. The other one is a pentose, 5 carbon atom, this one, and the other one is the hexose. Okay, 6 sides. Okay, like that. Okay, copy only the structure. We will evaluate. Okay. Double bond, and then here double bond, and then here double bond. But in the side, the double bond will disappear because this contains a lot of attachment. This guanine. Okay, ganon, chararat. Okay, so in the top portion we have oxygen. The double bond, there's also a double bond of oxygen outside. There's a difference for the guanine. Okay, like this, double bond oxygen. Okay, and the center, again, in the corner, because there's no double bond, again, by technique and H. Right? Memorize that completely, and even though it will be rumbled, and then you can understand, and you will adjust, something like that. Comparing to memorizing only, then you don't understand, then you will be lost. Okay? This one, there's a double bond, right? So you will put, every double bond in the corner must have nitrogen. Okay? Like that. Okay? So here, the corner has no double bonds. As I said, an H. Okay? So there's a double bond also on the top portion here. So that's nitrogen. Okay? So in guanine, there is only one side that has no covalent bonding with other element here. That's also one of your identification, so that you will never be lost. In the guanine, there's only one area of the zinc. Okay, meaning covalent, there's no attachment of other element in the corner, something like that. Okay, so this is only the exemption. Adenine and guanine are having 2 polygons, the pentoses and hexose. The procedure. Okay, it's already correct, right? I write already the 5-1. 1. Okay, for thymine, T with the first letter of the, nuclear base, put inside the square. Okay, adenine is A. What is the structure of a ribonucleic acid? Okay, I will show you now. Now, in terms of stability and structure, the DNA is more stronger than RNA because of their functions. Because the RNA will always only manufacture, but store, will try to transmit, will try to, express, like that, and everything is only one stronger than RNA because it has more functions. And another reason also pairs. For example, guanine plus cytosine genes. For example, an infant, a child, or an adult or anything, we can test that through DNA. Okay? So if it doesn't have a good match, then problem. Okay? For the pairing. Okay? So it should match the because it is a study of hereditary traits or inheritance through genes. RNA and DNA will be equal. So it will form like this. Okay? If there is wrong in the specific sequencing, it will never match. That means nucleus pairs. That means your sperm cell, semen, or I think anything they can do it, but I'm sure it's sperm cell or semen. I think so, or egg cell. It depends on the situation. Because I'm not a doctor, okay, like that. But they have to test sample from you, could and the laboratory test for the DNA test. The principle is like this, okay? The one I told you. Now I go back to protein synthesis, and there will be no genetic information that will be produced, okay, by the RNA, okay, to be transmitted by the DNA in the human, cells of the body of a living organism. This is not only applicable to the human being, but it's also applicable to all living organism. For example, a cow, like that. A cat, a rat, they have also their children like that, right? So you could also identify that this not only because there will be no disputes of animals. Okay, it's not controversy for animals. Okay, it's controversy only for human being. Okay, like that. But actually, it's being there. Okay, like this. But I'll try my very very best. How can I do this? Oh my gosh. Two. Oh my gosh. Four. Ganon. 5. Ganon siya. So, 5. Why? Because of the principle of pentosis. Okay? 5 carbon. Something like that. Okay, it's a component of nucleotide based on our definition at first in the introduction discussion, right? A nucleotide is composed of nitrogenous base, 5 carbon atom pentose. 1, 2, 3, 4, 5. Okay, like that. Ribonucleic acid. So let's do this like this one. This one. Or a spinal column of a human being. Ciao. So there is only, there's only one helix, this one. One helix backbone for the RNA. Like this, okay? And now, we need to locate the nucleotide base, okay? Which is the uracil, adenine, cytosine, blah, blah, blah. So we will put that here. For example, this is the adenosine. It's very difficult to if you will never see in the laboratory, right? This is the nucleotide bases to produce a protein synthesis, right? So let's say, for example, this is a okay, cytosine. I'm confused with the cytosine with the amino acid. Adenine. Example only, like this. Let's say like this, okay? So this is adenine. And then we have the guanine. Let's say guanine, okay? Guanine. And then we have RNA based on the theory. So uracil, like, okay? Uracil. Okay. So for example, this is the structure that here on the first curve. Okay, here. Okay, thing. Okay. React to the other edge, it will be compact now in the DNA, which is the 2 helix backbone. Okay, like this. And it's very difficult to pattern this illustration only, okay, like this. Okay, this is an example. Oh my gosh. Let's say this 3, okay? No, this 4, sorry. like this four. Okay, let's see, this one is the adenine A. This one is the guanine. Look at my paper. Okay, cytosine, like that. Okay, see? And then we have this one as a uracil RNA. There's no thymen, right? Okay, like that. Okay, now we draw, this is RNA, do the DNA. Two, so we need to pair that one. Okay, opposite with each other. Two like this and opposite with each other, meaning they react with each other as a two helix backbones. Okay, DNA. Okay, very difficult to draw, really. So like this, kaloka, okay. Grabe. If I have a printer, it's easy, right? We can print like this. Okay. So this is the 1, 2, 3, 4, 5. This is the one helix backbone in the RNA. Okay. Plus another one like this in opposite direction. So like this. Opposite will be here. See? And like this. Charara. Like this. Oh. Just like a chain. Really. A metallic chain. Charot. Okay? In lock out and tag out. Okay? Metallic siya, or it is a chain of nucleic acid. The definition of, this nucleic acid is gonna be like that. Nucleotides. See this one. Opposite direction, like this, together to become two helix backbone. Also, the nucleotides each other like that, blah blah blah, so and so. Here, example, this one. So the nucleobase will pair with each other, backbone. They reacted with each other to form the pairing. Okay, so like this until going down. Okay, so they're paired now. So that's the time all these nucleobases are compatible in the protein synthesis. Okay, like this. Okay, until down like this, just like a spinal column or a skeletal, something like that. So if this adenine will react to, let's say, guanine, according to their appropriate and specific behavior that we will clip together, okay, so until down, okay, pairing is not compatible, okay, that's the time there will be a problem for the DNA, okay, like that, okay. That's the logic and principle behind. So if they are keeping with each other, meaning to say they are compatible with each other, of course, okay, or else they will repel if they are not compatible with each other during the reaction or the bonding, okay, like that. So this is gonna be the principle behind the DNA. So meaning to say, we cannot obtain the DNA test without starting first with the RNA. Okay? So if you put that in a process, RNA is the raw material. And DNA is already a product. Something or the is there to make protein synthesis, to get this sequencing, specific sequencing. You know the word specific, meaning it will never go wrong. It will be compatible with each other. If there's something wrong, then it will not be like that. You understand the point? Okay, it's very clear. So this is gonna be in the inside of the cells and outside of the cells. Okay, going back to since you already see the structures, going back to the specific definition of DNA, the DNA will encode, okay, like that. And the specific sequencing in this nucleobase of the DNA, okay, this will go to transmit or storing the code behaving properly, functioning properly to its, specific function like that. Oh my gosh. Okay. You understand the point now? Something like that. Okay, again, this DNA, there's always a genetic information. Okay, something like that. And then this specific sequencing in DNA of this one, nucleobase pairs, enables for the storing and transporting, okay, or living organisms, including the human being, something like that. So, okay, and the RNA, still specific sequencing, specific, meaning to say not random. It should be compatible during backbones of the DNA, like that. Okay? This specific sequencing, there's a difference, right? Manufacture or produce new protein. So meaning starting to produce new protein, okay, for this and frames and other chemical processes of all living organisms. So very important, right? Structures, the parts and the frames of all living organisms like this, okay? It's gonna be like that. Any questions so far about nucleic acid? Okay, if the question goes like this, now, which of the following is not a macromolecule? Ganon. A, phospholipid. B, protein. C, carbohydrates. All is not a part of forming classes of macromolecules. Okay? Something like that. So, a lot of questions like that also. Okay? So, I will give you an example of a question. I prepared also some questions right here. Okay, question number 1. What is the specific nucleobase of the DNA that will never go to RNA? So the answer will be thymine. Okay, like that. Because thymine is exclusive only for DNA. Okay, like that. Okay, so if this thymine will not react there, this specific role for DNA. Okay, RNA also. It will not manufacture, okay, misses, and then there's no coded genetic information if this RNA will not use uracil, because this uracil is a specific nucleobase of this RNA also. Okay, like that, and it will react there for others nucleobase, something like that. The story will be goes like that. Okay, compatibility, specific sequencing of this nucleic acid sequencing. Okay. So in short, meaning to say that this is a biopolymer, a large molecules, okay, large molecules or biomolecules, really very, very essential to all known forms of life. Okay, it has to do something, the functions of storing, expressing, creating, encoding, okay, like that of genetic information, which is specifically the nucleotides, RNA and DNA, which is specifically the nucleic acid sequencing, something like that, okay? And then, to state it specifically, that this sequencing of amino acids in a protein is determined by nucleotide sequencing of the coded genetic information of this DNA, okay, and RNA, something like that, okay, or protein. So meaning to say, if there is DNA or RNA, protein must be there always, okay, because that's their partnership. If there's no protein, there will be no nucleic acid. So it's just like that. So if this is a business, so they have partnership with each other, corporation, wherein the member is the RNA, the DNA are, corporation with each other, have shares tax, okay, before getting the profit, something like that. It's just the same story with inside our body, okay. Okay, you understand the point for this, okay? This nucleic acid. And now we go to the secondary metabolites. Okay. The last topic for the, oh my God, there's also one, phospholipids. Okay. Phospholipids, very important because that is the major component structure of the cell membrane of an animal or all living organisms in the earth, including the earthworm, the grasshopper, the beetles, okay, the what else? The butterfly, the fireflies, the moth, the monkey, the mosquitoes, all. Okay? The cow, the cat, the bat, the bird, the cat, the dogs, okay? The cockroach, all. Okay? So it's very, very important. Okay? The phospholipids. Because this is the cell membrane. Okay? All animals and living will have a cell membrane. So meaning to say, without fat also, FAT, we cannot survive because it will develop the strength of the cell membrane to be strong enough that it will not be destroyed or broken during the physiological process of different chemical substances inside our body during action, propagation, digestion, like that. So, it will become a waste inside our body and we will get sick. Okay, we will be having disease all the time, like that. Okay, so this is gonna be important also the phospholipids. That's why phospholipids. Okay, like you check that an alcoholic fatty liver disease, you need more phospholipids in your because fats is an example of phospholipids. Okay, the fats, the lipids, okay, hormonal steroids, okay, is part for phospholipids. That's very important also, okay, like that. That's why. And we discussed also the fat referring to the phospholipids. Okay, like that. And there's a relation also with the carbohydrates because there's a triglyceride, something like that. It's just like a playing a game and regulation, behavior with each other. They're helping with one another like this, complementary with one another. That's why if you have a disease in liver, other organs will be affected also. Okay, burden, your lungs will be damaged also later on, okay, to function properly, something like that. Okay, or if there's no therapy for that, medical therapy for that, so it will be connected with each other. If you have one critical disease, the other organs also will be affected because they are really interrelated with each other inside our body. That's why we need to study the physiological chemistry, okay, and the anatomy of our body. So understand it better, we need a solid background of biology and chemistry, okay, biological reaction and chemical reaction, okay, like that. Okay, so because it's very enjoyable, very uh, very good to listen if it's really an actual, right, rather than studying the chemical reaction that is not in the human being. Okay, it's not, for me it's not really that relevant. Okay, for me, for survival, you're studying only the chemical reaction, how to produce textile like this. It's not that really important to the process, the chemical reactions inside our body, in which we will make us alive for more than 10 years, 60 years, 65 years, 70 years, okay? So those are non-living thing, right? The producing of the rubber, producing of the plastics, producing of the cement, producing of the gas. So they are non-living thing, right? Non-living thing. There are two living things and non-living thing. They have no life. Okay. So whereas if you try to enjoy our family, chemistry, biology, and health science for ourselves, not on the process of any companies and factories, it is not really that, for me, it's not okay like that. Okay, thank you so much. Oh my god, I'm finished. Phospholipids, okay. All this I discussed a little bit, but tomorrow I will be discussing more about phospholipids. Okay, but I need to discuss also the secondary metabolites. Okay, take note: phenolics, terpenoids, and okay, phenolic, like that. Phenolics. So what are these phenolics? Actually, this is not important for the development of, the important really is for the, against any diseases and other, what we call this one, the one who will eat the plants, okay? Herbivore. Herbivore, okay? Eating plants, right? Organisms or herbivores, okay? So it will protect that one. So if it will protect that one, I love those programs, okay? Quiz bowl, international quiz bowls like that they will compete with each other. I love to watch science because I don't love too much with mathematics. So this lignin It's just really, I know, because we're not studying this because we're chemical engineers. We're not focused for the bio part. Is and then this is a very strong component for the cell membrane, something like that. Very strong component of the cell membrane. So lignin, like that. Okay. And the flavonoid have so many functions, okay? Prevent diseases, cardiovascular diseases, used as an antiviral agent, okay, like that. The lycopene in tomato, okay? And the flavonoid for preserving the leather, leather, Lether, or textiles, Taxtiles, my gosh, is the tannin, okay? Tannin is the flavonoid to preserve those textiles and leathers, okay? Like that, okay? My gosh. And another uses of flavonoid also, my gosh, ganon. Another function of sex will carry the pollen, right? In pollination process, right? Of the female flower. Okay, female flower, female flower. That's called the the stamen, right? And it will react to the male flower, which is the stigma. And upon reaction, like blah blah blah, so and so, it will produce, okay, the seeds, the fruits, like that, okay. Like for example, the bees will produce the honeys, like that, through the process of pollination. So pollination is very good to the plants because it can produce a very good harvest, like that, okay. And the seeds will be healthy, and it will flower to world. So that's the flavonoid function also, okay. And then it will also repel herbivores, okay. Most substances or chemical, biological substances or chemical substances in our body, biological substances or chemical substances in our body have bacterial decay point. And then this flavonoid time, like that, it will delay. Okay, something like that. Okay, so there's really a purpose for this flavonoid. Okay, I really want to check really the composition of these multivitamins. Okay, and any vitamins. I want to investigate. Wow, and I would like to okay, like that. So another thing also, we have the allelophatic. Okay, allelophatic is the same with the characteristics with the flavonoid, lignin, like this, blah blah blah. Okay, the function also secreted with this kind of allelophatic. Secreted, absorbed, right? The roots, and then the purpose is to to reduce competition, because there are times also that these plants will grow in abundance and then there's no space anymore, and they were also quarried, so there will be no competition anymore. Okay, this will prevent from individual plant to keep on fighting, just like us, we keep on fighting for applying for a job, and there's one only position, a lot of applicants will fight like that. The plants also is like that. They will be eager of stealing this nutrient, and so many plants will also steal like that, compete with each other. So the purpose of this allelophatic substance is this, to avoid competition for the nutrients, for the space, like that, okay, and a lot more. But this is responsible to produce a new healthy plant. For example, this is the plant, okay, plant A and plant C. So if these plants are having the allelophatic substance or compounds later on, okay, and this will reduce the competition of these four plants. So mean to say, they are enjoying equal benefits for the nutrients, fertilizers, spaces, like that. If these plants have adequate allelophatic substance, okay, you get the point? Oh my gosh. So new terms here is lignin, flavonoid, allelophatic, phenolics, alkaloid, and against diseases. That's their main function of all these terpenoids, this flavonoid, allelophatics, common, okay, phenolics, okay, like that, the lignin, like that, blah blah blah. And then because these are not essential really for the, for the development of the growth of the plant cells. Okay, like that. So just like the have enough or adequate of white blood cells, right, to fight against bacteria, okay, viruses, like that, to have a strong immune system for our disease mechanism system. Okay, it's just like that for the plants also. Okay, the analogy, the comparison, something like that. Okay, so if this white, and these are all the leukocytes, right? So these phagocytes will touch the viruses, okay, and the bacteria, and these lymphocytes will kill them all. So it's just like that also in the plants. You can have adequate also of white blood cells. Okay, not only the red blood cells, which is our hemoglobin needs iron. You know iron element for in order for the hemoglobin to function well, because he is the one, or this is to carry the oxygen, okay, to be transported to the bloodstream of different main organs. Okay, so problem also, okay, like that. So we need iron. So what are those foods rich in iron? Food rich in iron. Iron, Fe. Oh my God. Okay, it's in the water. Okay, water has so many minerals also. Potassium, iron. If you see the ingredients of that, okay, after like that, it's only had made us strong, just like the protein, the lipids, okay, like that, the nucleic acid, the vitamins, so okay, for our cell wall for inside our organs, like that. So it becomes strong if we have adequate cholesterol level. Okay, so if more than, will be not more than 100 milligrams per deciliters. Okay, like that. Okay, the threshold limit or value. I don't think so. What's the term? Because there's a lot, right? Threshold limit, value, like that. Okay. Okay, thank you so much. You can like, you can comment, and then you can share also if you want to anybody else that you think this is beneficial. Coming newest updates, specifically regarding occupational safety, health, and science. Okay, or health science or fitness, like that. Okay, it's gonna be health science. Okay, OSH plus science. Okay, like that. But in other situations also vlogging cooking, that's personal vlogs, okay, like that's on health. And then personal vlogging, okay, this personal vlogging would mean etc, right? I can do cooking, experimentation, I can do whatever personal vlogging also if I want to, especially exercise or whatever, okay, or eating moment galore, financial management, right? It is not part of OSH, but I did that one. It's not really related, but I want also to to experiment another topic that is not my comfort zone. Okay, like that. So I want to experiment. So much, bye bye everybody. God bless. Woo! Mwah. Talaga.